Okay, good afternoon. Today, we want to start really going over the nervous system and understanding the nervous system, at least the general overview of the nervous system, so that that when we discuss how the nervous system benefits the body as a whole and how other body systems are controlled and coordinated by the nervous system, that it's not so unfamiliar, or even to a point where the the nervous system is frightening or scary in its discussion. What we aim to do is to make the nervous system comprehensible, understandable, to where when you hear its relationship to the other body systems that we talk about over the rest of the semester, that you see that nice congruency of the different body systems with the nervous system. So let me go ahead and share the screen so you'll see what I'm seeing. And let's go to the PowerPoints, chapter 23 in the Salvo book. So the nervous system has two subdivisions. I mentioned it on Thursday. I'll talk about it again in brief. The central nervous system is the part of our nervous system that includes the brain and the spinal cord. Everything that's brain related, everything that's spinal cord related, it's central nervous system. So the brain is housed within the skull. It's protected by the cranial bones, housed within the cranial bones. The spinal cord then begins once the brainstem enters through the hole in the skull called the foramen magnum, which is part of the occiput bone. And then the spinal cord will run down a canal of the backside of the vertebrae, completely housed in bone again. And the housing of bone is designed to protect the brain and spinal cord from impact forces, traumas that could potentially impact the brain and the spinal cord. The brain and the spinal cord are completely surrounded by a covering. It's actually a 3-layer covering called the meninges. So the meninges then would be a part of the central nervous system. And within the meninges, the brain and the spinal cord are bathed within a fluid called cerebrospinal fluid. So at some point in our discussion of the central nervous system, we'll be looking at the brain, the parts of the brain, how those parts function to benefit the body as a whole. And then we'll be looking at the spinal cord, the parts of the spinal cord, where it starts, where it ends, and again, how it functions to help, the whole of the nervous system provide benefit to the body of the whole. So because the meninges cover both the brain and the spinal cord, because cerebrospinal fluid bathes both the brain and the spinal cord, they are considered central nervous system structures. The only real curveball, or the only real oddity that jumps out that ends up being part of the central nervous system, is the optic nerve to the retina. The optic nerve is actually considered part of the central nervous system. And that took me, as a student of anatomy many years ago, that took me a little bit of time to really appreciate, because I was not really a fan of calling the retina or the optic nerve part of the brain. And my, if you will, my evidence was, well, we call it the second cranial nerve, and cranial nerves are not central nervous system. Cranial nerves are outside of the brainstem. They're peripheral nerves. And so why do we call the second cranial nerve the optic nerve? And for all intents and purposes, it looks like it should be a peripheral nerve. But when you get into the histology of the optic nerve, when you dive into its microscopic architecture, you find that the cells of the optic nerve are the same as the cells of the rest of the brain and spinal cord, that differ from what we see as far as the cells that make up the nerves of the periphery. So even though it looks like it should be a separate cranial nerve, in reality, it develops like every part of the brain and is as vital and as really as delicate as most of the parts of the brain. So turns out that the optic nerve is going to be part of the central nervous system. But for now, and that's really because it's part of the brain, for now, let's just go ahead and think of the brain as its main parts when we talk about it. And when we do go over the optic nerve, I'll remind you again that it's actually part of the central nervous system, even though it gets discussed with the nerves of the peripheral nervous system. All right, so I mentioned the word peripheral nervous system. And again, those are the nerves that are outside of the brain or outside of the spinal cord, the nerves that come off the brainstem, the nerves that come off of the spinal cord. and become the larger nerves of your body that allow your brain to communicate with the parts of the body. So think of a nerve as a biological wire, and it's a wire that exists between the brain and the part of the body so that communication back and forth can occur. And if you remember, when the brain sends information down that wire, that's a motor instruction. It's a motor direction of communication, such that there's an expectation that the body part is going to carry out the instruction that the brain is sending to it. Whether that's going from the brain to the muscle to tell the muscle when to contract or relax, whether it's the brain going to the skin to tell the goosebump muscles to contract, or the sweat glands to at work, or the oil glands to work, whether it's going to the thyroid gland to make more thyroid activity happen through hormones. There's an instruction sent, so the direction of impulse is always from the brain to the body. That's a motor or efferent direction of contraction of stimulation, of impulse traveling. I've got contraction on my brain, I apologize. So the impulse is going to travel from the brain to the body. If it's motor or efferent, it's going away. But that same nerve has other tracks where stimulations around the body, the nerve endings of the body pick up those stimuli, and stimuli are just the Latin way of saying stimulations. And whatever it is that the nerve ending picks up gets transmitted to an electrical impulse and then sent directionally toward the brain. And that's what we call a sensory direction of electrical impulse or nerve conduction, or if you will, an afferent direction of nerve impulse. So sensory afferent, motor efferent. Let me move this to the top here. Okay, you might not see that at home, but I see it in my computer. So anyway, with the periphery, the peripheral nerves are all the nerves that are not brain or spinal cord. So the 12 pairs of cranial nerves that come out of the brainstem, the 31 pairs of spinal nerves that come out of the spinal cord, they come out and then they become the nerves of the body that you'll eventually learn is like the sciatic nerve and the femoral nerve and the radial nerve, the ulnar nerve. Most of the nerves are actually named for the bones that they run with. Some of them are named a little differently. Then we also have, for example, multiple nerves named for the same bone or region. So then we just have to add adjectives to how they differ. Sometimes it's deep, sometimes it's superficial, sometimes it's medial or lateral. But those are just words of description that help us tell like branches, for example, of a nerve that's a common nerve that branches. They both might still be called the same nerve, but one goes deep, one goes superficial. So this is the superficial nerve, the deep nerve. Some refer to it as superficial branch or deep branch, but then you're getting into semantics as to whether or not it should be named as a separate nerve or just a branch of an original nerve. So that's neither here nor there at this point in the discussion. The point being is that they're still cranial nerves, spinal nerves. They are the nerves of the periphery. All right. When you get to nerves, we have a tendency to talk about nerves that allow us to have somewhat voluntary control of our body that goes to our muscles and our joints. So, like, for example, if I'm talking about the nerve to the biceps brachii, all right? So the biceps brachii is the muscle here in the arm, the little muscle that, well, for me, a little muscle, but for some people, a big muscle where, you know, when I was a kid, I was like, look how strong I am, right? That's the biceps brachii. And for those of you that already had the first semester, hopefully you remember that's biceps brachii, or as I used to teach it, biceps brachii not attached to the humerus. But the nerve that allows the brain to operate that muscle is called the musculocutaneous nerve, which is, in my opinion, the worst, absolutely worst named nerve of the entire group of nerves in the body, because musculocutaneous means the muscle and skin nerve. And it seems logical that the muscle and skin nerve, if you're going to call it just the muscle and skin nerve, will go to all the muscles and all the skin of your body, but it actually only goes to the skin of your arm and to the muscles that make your elbow bend. Okay, so why it's not called the brachial nerve or the biceps brachii nerve, or, you know, something that's more related to the arm rather than skin and muscle, I can't tell you. You know, when I went through the chiropractic program, I'd already had some anatomy classes in undergrad. I'd had a year of anatomy with cadaver work. In high school, I took anatomy, and we were just given the names of the nerves, and you had to learn them or whatever. And when I was in chiropractic school, the anatomy instructor that we had at the time, his name was Professor Ellis, and he looked at us and he said, you know, when a nerve goes right into a muscle, there's really nothing wrong with calling it the nerve to the muscle. So the nerve to biceps brachii, the nerve to triceps, in the scheme of things, if the nerve is going into the muscle and innervating that muscle, well, then it's absolutely the nerve to the muscle. It just happens that, some nerves will go to multiple muscles, or, you'll have a bigger nerve that branches to this muscle, and this muscle. And so other anatomists were like, no, you need to know the name of the nerve. You can't just say it's the nerve to the muscle. But I think one of Mr. Ellis's approaches in doing that was he really had a greater emphasis on us wanting to know the muscles more so than the nerves. So it made us want to learn those muscles better, because if you saw the nerve coming into it and you didn't know the name of the muscle, you're more likely to learn that muscle. And it was all about repetition and familiarization. And it's a trait that I took with me into my teaching career, because I really thought that that approach that he had was very effective. So I do the same. Now, with that said, if a nerve goes to a muscle that you control, it's part of what we would call a somatic nerve. But there's a whole, if you want to call it an underground system of nerve, there's a whole set of nerve that governs us automatically, involuntarily, whether we're conscious or unconscious, subconscious, awake, asleep. This nervous system is governing us, and it allows us to operate and function, but it doesn't make us really fully aware of the fact that it's happening, unless there's something wrong, or unless we need to know, then we become aware of it. But for the most part, it all happens automatically. It's self-governed, and I'm glad I don't have to worry about it. I'm glad that there's a part of my nervous system that will speed up my heart rate and my respiratory rate if I need it sped up to meet the demands of the day. I'm glad that in turn it can slow down my body when I don't need all that activity. I'm glad that during my relaxed phase of the day that can govern my digestive system, so that the digestive system is more active, so that as I ingest food, I can acquire all those nutrients and get that delivered to the cells of my body, so that my body is ready for the next day. Sometimes I need to be in more of a go, go, go mode, and sometimes I need to be in a really chill, relaxed mode. And I'm glad that I've got a part of the nervous system that can govern that without my awareness, without me having to consciously think about making sure my body parts and my systems are congruent in that manner. So that's what we would call the autonomic system. And ultimately, we're going to talk about the autonomic using fancy words, sympathetic and parasympathetic. And again, all this is peripheral nervous system, not central nervous system. But when we use the word sympathetic or parasympathetic, we're talking about the division of the autonomics that's either going to burn fuel real quickly and get us out of a bad situation, or going to conserve energy so that, when we do need it, it's there. And there are phrases that we typically use for each. Like, for example, the sympathetic nervous system, and I'll go ahead and use my laser pointer on it. Let me get my laser pointer out here so I can highlight it. Let's just put the laser pointer on. Okay, so the sympathetic nervous system. The sympathetic nervous system uses essentially a chain of specialized nerve and clusters of nerve cell bodies that we call ganglia, to make sure that all parts of the body are connected. And if you go to a fight or flight mode in an instant, that chain activates everything to make you a very efficient machine. You will never be stronger, faster. You will never think more clearly. Time will never be as slow as when you're in all-out fight or flight. All right? Because this is your body. This is your body needing to get out of a harmful situation. So it's referred to as fight or flight because you're either going to fight, or you're going to flee, or do what you need to do to get out of that harmful situation. It is self-preservation to some degree, which means I need more blood flow to my body, so my heart rate goes up. I need more oxygen to my cells, so my breathing rate goes up. I don't need my digestive system active. I don't need my urinary system active. I don't need my reproductive system active. So they're all put on the down low. It's like being on a battleship. You could be the cook in the Navy on a battleship. And if there's no battle going on, the cook is in the galley prepping food, cooking for the sailors, making sure that everybody's well fed, because that's part of the job. But if there's an actual fight going on, the cook is not in the galley making food. The cook is manning a muster station or a med station or a gun, and part of the fight. Nobody's in the kitchen eating during an actual battle, because the focus is on the battle. And when you're in fight or flight mode, you become a machine because you're quickly using energy that's been stored up to get you out of that situation. Again, you will never be stronger, run faster, think smarter. I mean, time really slows down, if you will, because you're aware of everything. So much blood flow goes to your brain, to your muscles, to your skin, nerve endings to your skin are heightened, but you don't need saliva from your salivary glands. Your eyes are wide open so that more light's hitting your retina, but you don't need the stomach breaking down food that you've just ingested. In fact, your liver goes into sugar factory mode. Your adrenal glands pumping out adrenaline so that you stay on the ready, because adrenaline allows your cells to just bring sugar right into their intracellular environment. It's really a very impressive state of being, but it should only last for a short period of time. For those that end up in a prolonged state of sympathetic, well, it actually becomes somewhat hardened to being in that mode, and it's a little bit more challenging for them not only to get into full all-out fight or flight, but then to actually come out of it and get more into the opposite mode. And we do believe that prolonged states of sympathetic fight or flight nervous system is ultimately what leads to PTSD, the post-traumatic stress disorders, following domestic abuse, following for example, military personnel that are exposed to military battle, where they've been, in fight after fight after fight, where maybe they've seen colleagues and other soldiers die around them, or maybe they were injured, or and then they come back to a non-military state where they're just, their sympathetic system is not functioning as it's expected to, and they struggle to reassemble into civilian life. But we do believe that it's long-term sympathetic exposure that does predispose them to that. The other side of that autonomic nervous system is what's referred to as parasympathetic. And instead of saying fight or flight, I like to use 3 different phrases. For parasympathetic, I like to say wine and dine, rest and digest, feed and breed. So why wine and dine? Well, wine, a little glass of wine, dining, that nice relaxed mood, preparing for the evening, breathing slowly, heart rate slow, digestive activities up, urinary activities up, reproductive activities up. That's the old feed and breed, right? Digestion and reproduction. But rest and digest is really a good phrase, because you're in that calm mode, you're in that down mode, and that's when digestive activities can really increase. So we find that massage therapy, for example, does a fantastic job of helping people convert from that shift of the sympathetics of the day, where the hustle and bustle and the stress of the workday, and then as you transition back to the wine and dine, you get a little extra help with techniques like massage therapy. And so we do find that when you can get a client all out relaxed on the table, they might even have a little twitch, their whole body twitches, and then they go to sleep. That's what we call the parasympathetic shift, where the body, let's say that you're in a regular everyday 9 to 5 job. You would say it's a little bit stressful. It's, get your stress levels up. You're using your caffeine and whatever else. You may not be in all-out fight or flight, but you might be leaning toward that side of the autonomics because of the situation. But when all that's gone, and then we're in the other side where we can get into that rest mode, that digest mode, that dine mode, that wine mode, that feed and breed mode, that's very relaxing. So think of parasympathetic as in a hammock, on a beach, pina colada right next to you, seagulls in the background, and you're just thinking, this is the life. That's parasympathetic. Your body is in conservation mode. As long as that's happening automatically and you don't have to voluntarily think about all that, you will be very, very, very happy. We'll come back to all this stuff when we get into the peripheral nervous system. All right. So if you look at this picture, I'll zoom in so you can see it a little bit better. Right there. All right. So they've got the brain in green and the spinal cord in green. And then all those other yellow nerves to the face and in the neck and in the body, those are peripheral nerves, okay? So the central nervous system, again, housed in bone, housed within the skull, housed within the spinal column. The peripheral nervous system, not necessarily housed in bone, following bone, maybe parallel to bones or running deep to muscle. But in some cases, some are relatively superficial. In fact, this part right here, let me get where the hand is, where I'm grabbing the picture. That's actually very superficial as it comes in from the body into the thigh. At least this part right here, not the big thick sciatic nerve, but this part right here to the front. That goes right over the pubic bone. This is called the femoral nerve. And then it becomes the long saphenous nerve. In fact, I'll just tell you some of the names of these, and we'll hear them again. First, I'll come to the outside of the arm here. The outside of the arm is this nerve right here, which we see coming down and then running down the forearm. If this is the true nerve as it would be really drawn, and it doesn't make it into the hand, then that would be the musculocutaneous nerve. This one in here to the armpit is axillary nerve, but what happens is that you have one nerve that's going to run down the center here and come all the way into the palm, and that's the median nerve. The radial nerve is actually running more to the backside along the radius, and the ulnar nerve is not really drawn well here. This one may be a little bit better, but it comes in over the elbow and follows the arm there. The green is the spinal cord. We have a bunch of groupings of nerves that we see here. This is what this grouping here is called the brachial plexus. It's what actually gives off these arm nerves. These nerves are called intercostal nerves, because they run between the ribs. And then as we get down lower, we see the femoral nerve becoming the saphenous nerve. We see the sciatic nerve becoming the common peroneal, then the deep peroneal and the superficial peroneal, and the tibial nerve runs straight down the back. But this is not a very well drawn example. If we really want to see the nerves, they wouldn't be a very good example. But again, those are peripheral nerves in the yellow and central nerves in the green. Central nervous system in the green. All right. Your PowerPoints go to the neuroglia, but I don't like to start with neuroglia. Okay. All right. So what I'm going to do is I'm going to actually bypass neuroglia and come back to neuroglia. I want to talk about the neuron. And where is the slide? Well, that seems to be out of order. Because the myelin sheath and myelin, what? Okay, I guess my PowerPoint's out of order. That's not a big deal. All right. So before I start talking about the neuron and the parts of the neuron, let's get a little general. The nervous system is comprised of approximately 300 billion nerve cells called neurons. All right, one neuron is not very effective, but 300 billion neurons as a network can create the type of communication back and forth between the central hub of the brain and the rest of the body that's effective for the control and coordination of all body systems. The neuron is the nerve cell that can transmit an electrical signal. It produces an impulse, or if you will, it conducts an impulse that is transmitted at a high rate of speed. That speed allows information flow to happen within fractions of a second. We need to be able to respond to our environment. It is that simple. We need to be able to respond to our environment. Which means if there's something in the environment that's harmful to your existence, we need to be aware of it so that we can preserve, that we don't let it harm us to a point where we don't survive. Which means that we have to have reactions fast enough to the environment and its stimulations, its stimuli, such that we're not injured, such that we're not killed, etc. Not only do we need to respond to the environment, but the chemistry that occurs inside of our body needs to happen at rates quick enough to then be prepared to respond to the stimuli of the world. So we need an effective means of making sure that if there's a need to change very quickly, that we have the way to get information to the parts of the body that allow it to change very quickly so that we're not harmed. And for us, we're very visual. So we use a lot of photoreception, a lot of information from our eyes to tell the brain about what is in the environment. What do we see? What do we see as far as things in the distance, things close by? You know, are we seeing something moving at us? Are we seeing something staying still? We rely pretty heavily on our eyes, but there are people that don't have the luxury of the sense of sight. We do also rely on what we hear as far as sound, what we smell, which may actually be one of the more primitive sensations. In fact, I believe that the sense of smell is probably the first line of recognition of a potential threat, especially if you happen to be a prey to certain predators. Those animals, especially, they're keen on the scent of potential prey. So the sense of smell gives us information. The sense of sound gives us information. Again, we take a lot of visual information into our eyes. Our hands and our feet, the places where we're touching the world, give us information about the way things feel. All right. So all this information is being received, the information from the environment being received, and then impulses are traveling at a high rate of speed back and forth between the brain and the body so that you can always respond to that information. To give you an idea of the rate of speed, the nerve impulse conducts at its fastest at about 277 miles per hour. All right, so it's pretty darn close to 300 miles per hour. 277 miles per hour is the speed at which an impulse will travel from the brain to the body back to the brain, okay? 277 miles per hour top speed. So the cells that conduct these impulses, they're built to encourage that rate of speed. And when we come to look at the nerve cells and how they're built, there will be a sheath wrapped around the nerve cell, at least the tail of the nerve cell. And that sheath, which is fatty, it creates a type of insulation that makes the ion channels cluster to where there's no sheath, so that the impulse essentially appears to jump down the long line of the nerve cell. So it's considerably faster as far as the impulse. It's not just slowly moving along a line, it's jumping way down the line. And again, 277 miles per hour when you're, say, 6 feet tall from head to toe. I mean, how long does it take when you're traveling 277 miles per hour to travel 6 feet? And I would say that that's much, much faster than a second. So I'm not a mathematician. I'm sure I could look it up to see what the actual length of time is between the brain and the body part when it's traveling at 277 miles per hour. But the truth is I don't care. I just want it to work so that, again, my brain and my body are in sync, that I can control and coordinate all aspects of my body and respond to the threats of the environment. That is what nerve cells do. So when we get into the world of our body, we find that we are the culmination of cells. And I said it's Thursday, but I'll say it again. Mom's egg cell was fertilized by dad's sperm cell. Mom's egg cell provided 23 chromosomes. Dad's sperm cell provided 23 chromosomes. As those two sets of chromosomes came together, your unique genetic instruction was built. And then as the fertilized cell there divided, the next two cells had the same identical genetic code, which those divide into 4, and those divide into 16, and those divide into 32. And it starts to become very exponential. And at first, those first few cells, I mean, I don't know if it's the first few days or first few hours or whatever, but the division's happening pretty quickly. And each and every one of those newly formed cells with that genetic material are identical to each other. You could take them apart cell by cell, and if they have the means to grow, they'll each grow into their own person. Same genetic blueprint, but individual people. So identical siblings, identical twins, identical triplets, identical quadruplets, if they are identical, it's because they were one fertilized egg that split, and then each was so viable they became their own person, but with the same genetic blueprint in their DNA. That's very different than fraternal twins, where more than one egg was fertilized by more than one sperm cell, and those are just siblings growing at the same time, being born on the same day, because once mom goes into labor, she's going to have babies. All right. So fraternal twins would be siblings, not much different than any other siblings that with families. Let's say, for example, if I do my brother and myself, I'm 2 years and 8 days older than my brother. We have the same parents. So if we were to have a DNA profile, I'd have my father's DNA and my mother's DNA. He'd have my father's DNA and my mother's DNA. So we would show as fraternal siblings. No different than fraternal twins. We were just born 2 years and 8 days apart. Now, if my brother was a half brother and we had the same mother but different fathers, we'd each have paternal DNA, but we'd have different maternal DNA. Those are half siblings. And that can actually happen with fraternal twins as well. All right. Because if more than one egg is ovulated and more than one sexual encounter with different partners during the time that those eggs are ovulated, one father can fertilize one egg cell, the other father the other egg cell. Those twins are born at the same time. They are fraternal twins, but of different paternity, DNA wise. Okay. And it's a Maury episode, especially when mom doesn't want to admit that there was more than one person at the party or whatever. I don't know the Maury story, but anyway, I'm getting off track. Eventually, those cells start to differentiate. And what that means is they start to become the more common cells of the body. Some of those cells will become the nervous system. Some of those cells will become the musculoskeletal system. Some will become the skin, right? So they're going to differentiate, and you ultimately get over 220 different types of cells. Not different cells in number, different types of cells. That if you looked under a microscope, this cell will be different from this cell will be different from this cell. And the very important ones that start very early on are the cells that become the nerve cells. They form into a tube called a notochord. And then that cord will eventually divide, or not divide, it will form a crease, and then it'll fold in on itself, and you'll start to see the beginnings of the spinal column in the nervous system. All right. So by the time the first trimester is over, 12 and a half, 13 weeks into pregnancy, there's not only been implantation into the uterus, but there's been a lot of differentiation of tissue types of cells that are going to become the nervous system, that are become the musculoskeletal system, that will become the skin, et cetera, et cetera, et cetera. All right. After the first trimester, there's a lot of growth of the body systems. And then as you get into the third trimester, mom's body's really starting to change. Baby's getting exponentially bigger, and delivery is on the way. After 40 weeks and 3 days on average, or 9 months, if you want to say it that way, baby will be born. And because of the big brain that human beings have, our offspring are born early. So they're fully dependent on mom for the better part of the first year to 2 years of life. They're not going to pop out and start running around a day or two later, like you might see with, horses or cows or other mammals on the farm, that even puppies, within a few days, they're just moving around, all joyful and playful, but they're smaller brains. They can be born later. They're longer in the womb. But the human brain and the human head is pretty big. In fact, when baby's born, baby's body and baby's head are pretty much the same size. And then baby's body will really grow. And there's growth of the brain and the skull also, but not quite nearly as different as the rest of the body grows. But with that said, the nervous system will eventually be a number of cells. 300 billion of them are the nerve cells called neurons that conduct and transmit electrical impulses. But neurons are not the only cells of the nervous system. The neurons are completely dependent upon a family of different types of cells that are responsible for making sure that the neuron is capable of being efficient at conducting impulses. So the way I like to simplify it is this. I want you to think of a neuron as royalty. I want you to think of the neuron as a prince or a princess. And that prince or princess isn't going to do a thing for his or herself. Okay? When that prince or princess wakes up in the morning, the royal entourage takes over. So the prince and the princess, they won't bathe themselves, someone bathes them. They won't dress themselves, someone dresses them. They won't feed themselves, someone feeds them. In other words, they are so royal that they just exist with constantly being tended to by other cells, and all they have to do is simply conduct an impulse back or forth, one direction or the other. And as long as the other nerve cells are feeding them, cleaning them, protecting them, bathing them, clothing them, whatever, then the neuron's going to be a very functional, happy cell. So originally, the scientists that looked under a microscope that saw these other cells around the neuron thought that they were cells that held the neurons together. So they called them nerve glue, neuroglia in the Latin, nerve glue. Neuron itself does come from, let's see, neuron, I believe, is a Greek word. Because if I remember nerve or neuro, what was the original etymology on that? Let me see if I can find that real quick. Neuro etymology. Yeah. So neuro comes from the Greek neuron, which was used to describe two different things, a nerve or a sinew, which is a tendon. Because essentially the original anatomists saw these things as cords, and the old term for a cord was a tendon. So because the nerve is the ropey part that was visible, as they broke it down, they saw that it was made up of parts. So the part making up the nerve was the neuron. The neuron as a cell is relatively large. There actually are different shapes to neurons, different, if you will, designs of neurons, depending on where that neuron is found in the nervous system. But regardless of the shape, as neurons go, they really pretty much work the same way. The reason why they work as they work is because their cell membrane, called their neurolemma, which just means their nerve sheath, it is dotted with what are known as channels, protein channels. And what that means is that when you look at the makeup of a cell membrane, it's this fluid membrane made of fatty acids. It's oil. Okay, we'll say it that way. And there's two layers of this oil, the outer layer and the inner layer of the oil, and the layers are held together by the inorganic substance known as phosphate. So cells in our body are made of organelles, and one of the organelles that we talk about when talking about cells is the membrane that surrounds the cell, called the cytolemma for a typical cell, called the neurolemma for a neuron, but it's a phospholipid bilayer. It's an oil-based membrane. It's not a wall. It's not a plant cell that has a cellulose wall, which is a sugar structure. It's a thin membrane that's made of oil. And the reason why oil is important is because the majority of what's inside the cell is water, the majority of what's outside the cell is water, and the oil membrane will keep the water separate so it's not just moving back and forth between the outer environment and the inner environment. Because if you know anything about oil and water, they don't mix. The oil won't just diffuse within the water and spread out. It will stay as part of its oil structure. So the membrane itself is a way to keep all of the internal content of the cell inside and all of the external world outside of the cell outside. But it does have channels. It can let things in and out of its environment. And some of the channels that are very important for nerve cells are what are called sodium and potassium channels. They have little proteins that have a closed shape. And because of their design, the protein itself opens a little bit, and then because there's more sodium outside the cell, the sodium starts to trickle in, and it slowly trickles into a point, and then all of a sudden the gate opens, and then it floods in. Once it floods in, there's this exchange of potassium that's inside the cell going out and sodium coming in, and it completely changes what's called the polarity of the ions along the surface of the cell. In English, it makes for a static charge. It makes for something that's electrical. And because of the way those gates are lined up along the membrane of the tail of that nerve cell, the gates start just flooding open, and they open down the length of the tail, which means that the impulse is going to start being generated. The static charge is going to start moving, or if you will, jumping down the tail, following the openings of the gates. Now, it probably doesn't make any sense at all what I'm saying right now. So when we finally do get to how a neuron transmits or conducts an action potential or an electrical impulse, and we see those gates, then I'll remind you again, that's what I was talking about. Even if you don't understand it, know this. Once that electrical current is on the neuron, has created enough threshold of electricity to start that current, then the static charge is going to move down the tail of this nerve cell. If it's moving toward the brain, it's an afferent direction of impulse traveling toward the brain. If it's moving away from the brain, it's a motor direction of impulse traveling to the body. But the point is, it's an electrical impulse. It got generated, and now it gets propagated or pushed, if you will, so that it shoots down the tail, again, at a top speed of 277 miles per hour. Which means once it gets to its final destination, which is the end of the nerve, the end of the wire, it will release chemicals between the nerve and the tissue it's trying to innervate. Those chemicals, are called neurotransmitters. And then the receiving cell has receptors that bind to those neurotransmitters, which are chemical instructions, and then the inner environment of that cell changes to meet the instruction of the neurotransmitter. So, in order to understand all this even better, know this. When one cell communicates with another cell, it happens because of chemistry. It happens because of chemicals. So when one cell wants to affect another cell, it releases chemicals, and then those chemicals bind to the receptors on the membrane of the receiving cell, and then that cell changes its activity. All right? So the question is how can you get chemicals from the brain to the receiving cells? Well, the brain bypasses the chemicals. The brain creates electrical impulses that then target the ends of the nerves where the chemicals are stored and then released to the receiving cell. The nervous system has learned how to use electrical impulses for high rates of speed to ultimately release chemicals into the spaces between the nerves and the tissues, to where those chemicals can then quickly change the activity of the cells. Now, there's a whole nother system in the body called the endocrine system. The endocrine system dumps chemicals into the bloodstream and just lets the blood carry those instructions around to the cells of the body. So it's going to take longer for those instructions to be carried out, because it takes longer for the instructions to get to the target cells. The nervous system is like having a hotline directly to the cells you want to affect. All right. But the endocrine system is a PA body-wide, public announcement of here's what we're going to do. And then the cells that are the target cells respond when they're basically when they get the instruction from the blood. So if a lot of this sounds like I have no idea what he's saying at this point, that's okay. There's going to be a lot of repetition over the next 2 lectures. But today, what I want to do is focus on the cells of the nervous system that are not only the neurons, the VIP royalty cells, but the entourage of nerve glue, the neuroglia. So let's talk about the neuron first. There are, as I said, different sizes of neurons, different lengths of neurons, different shapes of neurons, but they all function the same. They all take in information from their receiving ends into the cell along the tail of the cell, where the end of the tail will connect, or if you will junction with another nerve cell or another type of cell in order to then communicate with that receiving cell. There is a very big discussion of what is electrical as an impulse traveling along the axon or the tail of the neuron, and then the releasing of the chemicals for the communication between cells. There is a chemical communication between neuron to neuron. There's a chemical communication between neuron and muscle, between neuron and gland cell. As long as it's one cell to the next cell, it's a chemical communication. There are some connections that are not a chemical connection, but if you will, an electrical connection. Those are much more primitive. And essentially, it's just like the 2 parts fuse together and it just continues as if it's one part. But we won't see much of that. So if you don't ever remember that, that's fine. So the nerve cell called the neuron is the VIP. It's the royalty. It's the cell that is hopefully going to be around a long time, because it becomes part of a network that the nervous system uses for the regular flow of information to and from. If nerve cells die and they're in the central nervous system, they're not replaced. If nerve cells die in the periphery, they may be replaced. If nerve cells are injured in the central nervous system, they're gone. They're not regenerating or being replaced. If nerve cells are injured in the peripheral nervous system, there may be a full recovery of newly outcropping connections with the peripheral nerves reconnecting to the tissues following injury. So peripheral nerves are better at repair, but central nervous system nerves and nerve cells, not so good. But they've been with you since they developed, which could have been in the womb, could have been during childhood, could have been later teenage years, but they're going to be with you until they die, and they will die. I have read as early as after the age of 21, nerve cells, especially in the brain, start dying. 25, I've seen. But certainly by the time a person's 40 to 50 years old, the neurons of the brain are dying. You're not really seeing more being made as if when you're in your younger years. I came across one resource, some kind of encyclopedia of Encyclopedia Neurologica or something like that, and it claimed that after the age of 21, that neurons in the brain start dying at about 1 million per. I don't think it was 1 million per year. I think it was 1 million per day. And I remember thinking that seems like an awful lot, because that's 365 million a year, which means that in 10 years, it's 360. Yeah, that can't be right, because you only have 300 billion nerves. It must have been about 1 million a year. And I thought, okay, well, if you live after 21, if you live another 80 years to be 100, then that's going to be 80 million neurons of your brain that die. And I thought about it. Well, if it's 100 billion in your brain alone, and you're not even at 100 million dying, well, that's not too bad. But maybe it was a million per day. I don't know. I was trying to do the math in my head. Hopefully, we don't lose 100 million neurons, or sorry, a million neurons after the age of 21 dying a day. I hope not. Your brain is made up of roughly 100 billion neurons, and there's about the same number of neuroglial cells, which means there's another roughly 100 billion neuroglia cells in your brain. So your brain has, between the neuroglia and the neurons and the blood vessels, it's a few hundred billion nerve cells, a few hundred billion overall cells. So I hope that they stay around 100 billion, because the more neurons that stay with you the greater part of your life, the sharper you'll be as you get to those golden years. All right. Nerve cells, neurons. There are really two major neurons, three if we really break it down. We have what are called motor neurons. Again, they directly send impulses from the brain to the body. We have what are called sensory neurons, and they send information from the body toward the brain. And within the central nervous system, especially the brainstem and the spinal cord itself, we have what are called interneurons. The biggest difference between interneurons and sensory motor neurons is that interneurons are not myelinated. What that means is they don't have a myelin sheath that allows for a high rate of speed. So when nerve impulses travel on the interneurons, it slows down a bit. To give you an idea, the non-myelinated neuron, like the interneuron, the impulse travels 1 to 3 miles per hour, not 277 miles per hour. So 3 miles per hour to 300 miles per hour is a pretty significant difference in speed. Don't get me wrong, 3 miles per hour, when the thing is, shorter than that in total length, 3 miles per hour doesn't take very long. But almost 300 miles per hour is even faster. So, but they're non-myelinated. The other thing is that neurons can run the length of a nerve, and some nerves are pretty long in the body, and that means that you have cells that are running that long. The sensory neurons pick up information from all around, send the information toward the spinal cord, where the sensory neuron then connects to an interneuron. The interneuron is not only relaying to other sensory neurons that go up toward the brain, but the interneuron is also connected to a motor neuron that goes right back out to the body part. And the purpose for that is that if there's enough sensory input, an overwhelming amount of sensation coming in through those neurons, instead of waiting until the information is processed by the brain, the connection of the interneuron to the outgoing motor neuron, it'll just fire the motor neuron without any higher center processing, not even knowing what it was, just knowing it was stimulated, and there's a motor response. It could be too much pain, too much heat, too much stretch, too much cold, too much tickle. As long as it's too much of any stimulation, the interneuron allows the output motor instruction to respond to it, a reflex, while still relaying information in your brain. And so when we get to the peripheral nervous system, when we talk what reflexes are, which is the simplest form of nerve activity, it's because of the interneurons. When you discuss the neuron itself, and the neuron is as far as cells go, you're trying to learn these parts. The classic neuron that's used is a motor neuron, not a sensory neuron. The difference between a motor neuron and a sensory neuron is that the sensory neuron has nerve endings all throughout the body that immediately go into an axis. And then the cell body itself is out to the side of the axis that then continues on toward the what we call the terminal end of the cell. A motor neuron is very different because the motor neuron body is the beginning of it with its receiving processes all around it. And then it has to take everything in until it meets a threshold, which then uses its long axis to send information away from the cell body and out away toward the body. All right. I think that none of this is making sense. At least it's not making sense to me, because in my mind, I'm visualizing a picture of the nerve cell while talking about the parts, but all you see on the screen are the words. So what I'm going to do is show you the words, and then we're going to look at some pictures. Okay. So all neurons, and let me even do something here. I'm going to draw. So you have motor nerves with neurons, and motor neurons will have a cell body, right? And the cell body is going to have all these little tree-like processes sticking off of it. It uses the Greek word for trees called dendra. These are little trees, or dendrites. Okay. All these little branches from the trees. These are dendrites. Information flow is always going into the motor neuron through the dendrites. Okay. The dendrites are picking it all up. I probably should have done that a different color. Okay. So the neuron has a big old nucleus in it. It's one of the larger nuclei. Now, nucleus is Greek for seed or kernel, because when scientists first started looking under microscopes, they could see this big thing in the middle of many cells, and they thought that they were the seeds of the cell. So they called them the seeds, using their words, which is nucleus. There are other organelles in there, but the nucleus is pretty big. So the main part that I drew is the cell body. These dendrites, or little trees, are all around it. And notice that it's kind of coming to this funnel end. This is known as the hillock of the cell body, sometimes called the soma. And then it becomes a long tail. The tail is its axis, or its axon, if you will. Axon just means little axis. Okay. And then at the end of the tail are smaller little branches that bulb. These are the end bulbs, terminal bulbs. And they're connected to something else. It could be another neuron. It could be a another neuron, like I said, or maybe it could be a muscle cell or a gland cell or whatever it's connected to. The impulse is traveling in this direction, away from, it got picked up by the dendrites through the cell body, and then traveling down this tail. Now, this is your typical neuron. If this is in the central nervous system, there are little cells out to the side that will then reach and enwrap this thing with a little bit of space. And then it'll reach over here and wrap that over there. And then there might be one over here that comes down and wraps this right here. And then either way, they're on the outside. Okay. Another one right here would wrap this space. And there'd be another one and another one. Okay. These are what are making the myelin sheath that allows the electrical impulse to jump between the gaps. These are types of neuroglia. That's if it's the central nervous system. If it's the peripheral nervous system, then it won't be out to the side. They will be directly on them. There'll be one there, There'll be one there. There'll be one there. All the way down this axon, all the way down this little axis, the axon. In the periphery, they just surround them like jelly beans. And again, the impulse will travel to that joint, that junction, that it'll look like it's jumping all the way down. These, like the blue ones I drew before, those are called the cells that make the myelin sheath, the fatty sheath that allows that high rate of speed. Okay, oh, some chat stuff. Terminal end bulbs or synaptic bulbs, 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 end bulbs. I'll show it all to you. Yep. Okay, so that is, and by the way, everybody, this is a motor neuron. Well, that didn't work. Motor neuron. The hell? I don't know why it does that. I still haven't figured that part out. All right, so this is a motor neuron. Okay, and again, I'm going to show you real pictures of this. This is just my computer mouse drawing version. If it was a sensory neuron, it would have started like this. It would have started with nerve endings, some kind of nerve endings that all come in. Then there's the axon. Then there's a cell body out, and then the axon keeps going until it branches into the inner neuron in the spinal cord. So what happens is with the sensory neuron, the cell body with its nucleus, which doesn't have any trees on it, no dendrites, it allows the information flow to essentially bypass the body and get to the spinal cord faster. So it still has Schwann cells or myelin producing cells on there. But again, it doesn't have the same, its direction of impulse is toward the brain, whereas this motor one is away from the brain. So the brain would be over here. We'll just say brain. One of these days I'm going to invest in a actual pen and I can write it instead of having to click buttons with moving a mouse because it's bad. All right, I'm just going to show you all this because my artwork is not, it's not for sale, sorry. All right, let's do it. That's right. We're just going to show you. Let's exit out of that. Neurons. Here's a neuron. Okay. Does this look like the cell body I mentioned with its big old nucleus in the middle? Look at all those trees, those little trees, dendrites. Dendra, the Greek word for tree. So dendrite means little tree. And each of those little trees has a number of little branches, and each of those branches can connect to some other neuron or something else. So if, let's just say there's 1, 2, 3, 4, 5, 6, 7. All right, so 7 what look like trees, and each tree has 1, 2, 3, 4, 1, 2, 3. So each of the 7 has roughly, we'll just say 20 to 30 branches. That means there's 7 times 30. That's 210 potential branches of things connecting to it. All right, and again, I don't know what the real numbers are. I'm just throwing out there based upon this drawing. So that means you may have a few hundred other neurons connected to this neuron. And as long as the information is coming into these dendrites, it'll summate or collect, come together and add up as electrical information inside the cell body until it all gets to this point right here, the beginning of this long tail. The tail is called the axon. And the information will travel down the axon toward the terminal branches where you have end bulbs. Do you see these little round bulbs? The reason why they're bulbs is because this is where the chemicals are stored, the neurotransmitters. Because if you remember what I said earlier, when this cell goes to communicate with the next cell, it's a release of chemicals. Information between one cell and another cell happens through chemicals. It's called a chemical connection, a chemical synapse. Unless you're British, then it's a synapse, a chemical synapse. The connection of the neuron to whatever it's connected to is a junction point called a synapse, and the information flow is through releases of chemicals. So with this neuron, we have the cell body, sometimes called the soma, S-O-M-A, with a large nucleus in the middle, okay? There are other organelles in there, lots of ribosomal networks, endoplasmic reticulum. It is a cytoplasm environment, lots of actin and myosin proteins, which allow that cell to actually move a little bit. Yeah, it's a very busy internal environment, all right? But the nucleus is large and at the center. Once information comes in through the trees, in through the dendrites, all right? Pretend for a moment, if you need to, stand up and you can do this. If you're a neuron, your hands, your arms are your trees, and your hands are the branches of your trees. You would be receiving information that would travel in through your arms and then into your cell body. And if you put your legs together really tight, those would be your axon, and the information flow will go through your body and then out your legs down towards your toes, where your toes are connected to something else. But it would be one leg, one set of toes, not two. All right. So in this particular picture, we see this neuron here, that neuron there, that neuron there, that neuron there. And look at how each neuron's got its axon. And some axons are connected to some dendrites of other neurons. And then the axon is surrounded by these little jelly bean looking things. But when you look at it, you actually see it's almost like somebody took a long piece of rectangular paper and then just wrapped it. And this is actually a very good thing for the cell and the axon. This is called the myelin sheath. And what the myelin sheath does, it allows for a lot of surface area in a very condensed space. You reach full myelination in your mid-20s. And it's part of the reason why we're so, well, physically strong and fast and active in that decade, between 20 and 30 years old. If you look at many professional athletes that use their body, not athletes that use equipment. I'm not talking about race car drivers or things that like golf. I'm not talking about that. I'm talking about athletes that are runners, track and field, they are in their prime between 20 and 30 years old. And a lot of that has to do with the way nerve conduction occurs because of the fact that they're at peak myelination during those years. And then after 30, the myelin sheath starts to thin out a little bit. And so nerve conduction's a bit slower. So we're fastest, strongest, et cetera, in that decade between 20 and 30. Maybe starting a little bit before you're 20, 16, 17, 18, but certainly after the age of 30, things, maybe 35-ish, things start to slow down. Look, it's very odd to find a 70-year-old person competing with 20-year-olds in Olympic events. But perhaps there is a 70-year-old out there that's really still, peak myelination. And my guess is that individual is going to look 20 years younger than they really are. So we'll not worry about that. What we'll worry about is that if you look at a neuron, and I will have you look at a neuron, and you want to discuss the parts, this motor neuron has dendrites that take information into its cell body, and because it's built a certain way because of the genetic material in its nucleus, and it's provided with all of its resources that allow it to function through, again, the instructions in its nucleus. It will allow that cell to function to where it can ultimately transmit an electrical impulse that's going to start traveling down this tail. And because of the myelin sheath, it cannot travel. The thicker it is, the less it can travel through that zone. So it has to jump it to this gap, and then to that gap, and then to that gap, and that gap. And by the way, the gaps that I'm talking about, those are called the nodes. The nodes, N-O-D-E-S, the nodes of Ranvier, R-A-N-V-I-E-R, Ranvier. Let me show you some video of neurons and electrical impulses traveling down neurons. So let me close that image, and I'm going to go to some video here. Neurons. And there's a claymation one I want to show you, and then one that shows you a myelinated versus a non-myelinated neuron. That's a synapse. I have no idea what that is. Mixed neurons. Weird. That's a real neuron. Look at the dendrites developing outward. Time lapse, 12 seconds of that. That's pretty impressive, the growing of that neuron. So this is a grad student out of Stanford. She's got some pretty good videos on neurons and neuroglia. Neurons of nerve cells, 8 seconds of what it might look like. Can you see the way there's impulses just flashing, flashing, flashing all over the place? Neuron 3D animation. See that's the axon that's attached to other neurons, and the impulse is just traveling down the axon. But there's a synapse. There's one connection to another. All right, but that's still not what I'm looking for. I am looking for inside the brain. No. You would think that by now I would have got my own little set of videos saved, but I kind of enjoy looking at these to see if there's new videos I haven't seen. So that's why I've never done it. Let's try adding something here. And if that doesn't work, I'm going to change it to myelin because I know I'll find it there. There, that's the claymation one. So I want to do this one first because I really like the way she talks about it. So here we go.
I'm a therapist who goes to therapy, and some people think that's strange. The thing is, though, therapists are human beings just like you. If you've been thinking about or
understanding the properties of the neuronal membrane is essential. But understanding just these properties isn't sufficient to give us insight into collective behavior of the billions of connected neurons in our brains. Luckily for us, we can approach neuroscience at many different scales and levels of analysis, and we don't have to confront the full complexity of everything all at once. That's what we'll be exploring throughout the rest of this course as we slowly go from our understanding of single molecules, such as ion channels, to the electrical behavior of neurons, to their collective behavior in small circuits, and finally onto how they become organized in large functional regions of the brain. Let's start simple, though. Since we've examined one neuron, a logical next question is how do two neurons connect with one another? We'll first examine some basic cellular anatomy of neurons. So far, we haven't made too much of the fact that the majority of neurons are polarized cells. That is, they have one portion of the cell for receiving inputs and another portion for sending outputs. The parts of the cells that are specialized for receiving inputs from other cells are called dendrites. The word dendrite comes from the Greek word dendron, meaning tree, and as you can see, the dendrites have a branching, tree-like structure. A signal received by a dendrite is passed to the cell body. If there is sufficient depolarization of the cell body membrane to initiate an action potential, then an action potential is sent down the axon. The axon then carries the propagating action potential to another neuron. So what actually happens at the boundary between two neurons, between the axon of one neuron and the dendrite of another? This interface is called a synapse. There are two general types of synapses: electrical synapses and chemical synapses. Electrical synapses are less common in our own nervous systems, but they're simpler to think about, so let's start with them. Electrical synapses are basically pores between two cells that allow ions to pass through. They allow the passage of the electrical signal through to a neighboring cell without much fuss. It's not so different than just combining two cells into one larger cell. There are lots of reasons that nature might need synapses like this from time to time. They're fast and they allow cells to couple together with a high degree of synchronicity. But most neurons are connected together by a much more complicated structure called a chemical synapse. In a chemical synapse, rather than simply passing along an electrical signal from one cell to another, the action potential travels to the end of the axon and causes a chemical to be released into a very small space between the two neurons called the synaptic cleft. This chemical is taken up by the downstream neuron on the other side of the cleft. This chemical signal can cause the downstream neuron to depolarize its membrane, converting the chemical signal back into an electrical one. Or it can have other effects on the cell. This chemical step is slower than transmission across an electrical synapse, but it opens up an enormously diverse repertoire of different and more complex kinds of signaling. And synaptic function plays a critical role in computations performed by neurons. We'll spend the rest of this unit exploring the inner workings of chemical synapses, on our way to beginning to look at how networks of interconnected neurons give rise to behavior. We'll also look at the role of defective synaptic physiology in neurological and psychiatric disorders, and we'll see how synapses can be targeted by various psychoactive drugs and poisons. Finally, we'll wrap up by looking at how synapses can change over time in response to external stimuli, playing a foundational role in how we learn and remember.
I haven't seen any of those other ones, by the way, that she mentioned, so I don't know if she ever put them out there. All right, let me find the other one here. And that's Alzheimer's, that's that. So some of these are coming up because of like the pathologies that I show. And so it's my computer remembering. That one actually looks pretty good, but that's not the one that I was thinking of. Let me think. I tell you what, I'm going to play it because it looks pretty good. There's a lot of animation, but not a whole lot of explanation. But it gives you an idea of what's going on inside. Oh, look at that. Ooh, that's great. That's a nervous system right there. All those nerves. I'm gonna have to remember to show that one again. Ooh. All right, this is the one. It looks. This is what I wanted to talk. I wanted to show you. All right, here's this one.
Let's say I'm a biology student working on a research paper for school. As I work, I incorporate research from websites to support my ideas. It's
Schwann cells surround the neurons of the PNS. The axons of peripheral neurons are often surrounded by cells of another type, known as Schwann cells. These cells are important for providing support and nutrition. They also act as an electrically insulating sheath. The individual cells are interrupted by gaps called the nodes of Ranvier. Because of the insulating effect of the Schwann cells, signals travel along the axon in a series of jumps. This process is also...
I want you to watch how fast it ends up on the top one getting to the end versus the bottom one. The bottom one has no myelin, so it just slowly moves down. But in the top one, it's actually going jump, 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 jump. Latin and Italian and Spanish, salta means to leap or to jump. So saltatory is jumping conduction. So it's going bop, 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 and it happens to get all the way to the end much, much faster than what we see at the bottom. So let me hit rewind here. And again, I want you to watch how fast the top one is relative to the bottom one.
Because of the insulating effect of the Schwann cells, signals travel along the axon in a series of jumps. This process is also called saltatory conduction. The signal jumps from node to node.
So that was a neuron. So that was a neuron, right? What's this doing?
Signals travel along the surface of the neuron. Schwann cells surround the neurons of...
I'll let it go.
Signals travel along the surface of the neuron. Particles known as ions are responsible for this. Ions are electrically charged molecules or atoms. For a neuron, the major ions are sodium, Na, and potassium, K. They are unevenly distributed. Outside the neuron is high in sodium, and inside the neuron is high in potassium. At rest, the cell membrane is effectively impermeable to sodium. This creates a resting potential of about -70 millivolts across the cell membrane of a nerve cell. There are ion channels in the membrane of a nerve cell. When a signal arrives, these channels open and allow sodium to flow in. Potassium leaves through the cell membrane. The polarization now reverses and the signal is passed on.
All right, at this stage, don't worry much about the sodium and potassium, but that will ultimately be how the static is not only created, but then pushed. This is why potassium and sodium are so important in our diets. Eat your salty manners. Oh, your salty naners. Salty nanner, okay. Yeah, you don't want to have just straight water, but you know, if you're eating food and you're drinking fluids, you're going to get your sodium and potassium. But if you really are worried about depleting in those, Gatorade, sports drinks like that, they've got extra added values of sodium and potassium and other electrolytes to help with all that. Chicago lottery winner poisoned by cyanide after claiming him. Oh, that stinks. Okay. So when we go back and we look at the PowerPoints and we look at the neuron, this is what we see. That the neuron has its cell body. the dendrites bringing things into the cell body, and the long axon taking information away from the cell body. Cell body has the nucleus and all the other organelles. The dendrites are the little trees that bring impulses into the body, so they're around the body as intake branches. The axon is the outflow branch where the impulse is going to travel away and toward the next neuron or the next cell that it's connected to. It all funnels or summates where the cell body tapers to the beginning of the axon. It's referred to as the hillock. And then you start into the axon. As you get to the end of the axon, here's the fancy term, telodendria, okay, which just means the long telescope tree, the end of the tree, if you will, and that's the small little end bulbs, those terminal bulbs that are also known as synaptic bulbs because they're what form the synapse with the receiving cell. And inside that bulb are very specialized vesicles full of neurotransmitters. There's a roughly 300 neurotransmitters, 300 sounds right, around 300 neurotransmitters. I'm pretty sure it's not 30. I'm pretty sure it's around 300. I don't know why I would think the extra zero if it wasn't only 30, but regardless, the way calcium ions work is calcium ions will either flood into the bulb and push the vesicles into place to release them into the cleft, or the nature of the impulse being repetitive and the speed and the intensity will dictate larger or smaller nerve chemical structure, and it releases that appropriately. So the type of impulse that's coming down to the end of the neuron, the intensity and the speed at which it's coming down will dictate which neurotransmitters actually get released into the space. Then as those chemicals go into the space, because it's a fluid-filled environment, those chemicals diffuse, and many of them come into contact with their receptors on the receiving cell. And once they bind, it's a chemical interaction, and it can totally change the chemistry inside of the receiving cell. So here are the trees. Here's the cell body. There's the nucleus. There's the axon. And this axon is surrounded Schwann cell, Schwann cell, Schwann cell, Schwann cell, Schwann cell. This is what we would call a communicating branch, another collateral communicating branch. And then as you get to the end, there are your end bulbs. And look, there's muscle fibers. So this is a neuron that is innervating muscle. So the impulse will travel away from the cell body, down the axon to the muscle. That is a motor impulse because it's going away from the brain toward the body. This is a peripheral neuron because these are Schwann cells, not the cells that myelinate the brain and spinal cord. It even says nucleus of Schwann cell, unless you heard that one video where they say Schwann cells, but Schwann is how we say it in America. And look, in the distance, you see the way this axon is going, and it ends, its end bulbs are connected to another neuron. Well, that means that some of these end bulbs would be connected to dendrites or the cell body or whatever, however it connects. And then that can communicate with that neuron, which can then travel. So as you have numbers of neurons part of a same pathway or a circuit, if you will, they would communicate with each other regularly and often. That is your typical neuron. Now, just to give you an idea of the difference between the motor neuron and the sensory neuron, as I drew them before, I will show it to you this way. Motor versus sensory neurons. Efferent versus afferent. And let's find a picture, shall we? This is the motor neuron that we saw before. Dendrite, cell body, nucleus, axon, Schwann cell, Schwann cell, Schwann cell, Schwann cell, Schwann cell, end bulbs connected to muscle. This is the sensory neuron. Let's say that this is the receptor in the skin through the axon. No dendrites on the cell body. And then the last little bit before it gets into the spinal cord. When it gets into the spinal cord, it'll hook up to this thing. This thing will come down and relay either up toward the brain with other sensory neurons in the spinal cord or brainstem, or part of it might connect to a motor neuron because sometimes we need that for what's called reflex. This is a sensory neuron. Again, here's nerve endings in the skin. The information gets picked up due to touch or chemical or pain or whatever it is, temperature, and then it travels through the axon because the body is kind of out of the way. The nerve impulse very temporarily slows down before it continues on toward the brain and spinal cord. So no dendrites. This is a motor neuron where, again, dendrites pick up the information. There's your cell body, there's your nucleus, out your axon all the way down until it gets to its end stage. So those are actually pretty good little differences between sensory neurons and motor neurons. And I'm shocked that my two pictures are side by side just from the Google search. Usually I have to find 59 other pictures in between and go, oh, this is just like that other one. But that's the typical sensory neuron impulse going toward the brain, afferent, versus motor neuron. Hey, look, this is picking up touch, temperature, pain, chemical, vibration, tissue distortion, proprioception, which is positional changes. I mean, all kinds of sensory information in these different nerve endings traveling through the nerve toward the brain and spinal cord. So some type of environmental stimulation stimulates the nerve ending. It travels through the sensory neuron toward the brain. When there's a reason to change or adapt to that, then the motor instruction goes down and out, and then that muscle will contract or relax in order to move the body in response to the stimulus. So again, here is the motor neuron going to the muscle. Here's the sensory neuron from the skin going to the brain. Motor is efferent, sensory is afferent. Sensory afferent, motor efferent. Sensory afferent, motor efferent. SAME, same. If we were in Arizona, if this was Mesa Community College, I'd be like, look, just like where we are. Mesa, MESA. Motor's efferent, sensory is afferent. So that is the comparison. And by the way, this is the typical setup. This is the spinal cord. This is the motor neuron in red with the impulse going away and toward the muscles or glands or whatever. That same nerve has sensory tracts that come back in through the back part of the nerve into the back of the spinal cord. The green thing is the interneuron that is not myelinated. It's simply a go-between, such that if there's enough stimulation coming into this neuron, you see this bulb? It's called a ganglion. If we were to actually open up that little lump, we'd find all of the cell bodies of all of those neurons for that nerve that are sensory clustered in that lump because their axons are real narrow. Then there's a lump in the nerve where all the cell bodies are, and then the rest of it continues into the spinal cord, where it then relays with the interneuron, which goes up the back of the spinal cord toward the brain. As long as it's not overstimulated, that's the direction of flow, in and up to the brain, in and up to the brain, in and up to the brain. But if there's enough intensity along these sensory neurons, these cell bodies engage and they overload the information coming into the interneuron, and it will fire the motor neurons to respond to that signal. So you pull away and you move in response to that, even though you don't know what it is. When you put your hand down on a hot stove or you step on something sharp, you actually pull away before your brain knows that it hurts or whatever. And then your brain decides what to do thereafter. It's called the reflex arc. All right. So that's what I meant. So here's the person's sensory going into the spinal cord, the interneuron, and then the motor back out. And if that's a pinprick or if it's a hot pain, if it's something too intense, you actually have a motor response while it's relaying up to the brain, so that the faster you respond, the more likely you are to have self-preservation. Sensory in to the brain, motor out. Picked up, these are nerve endings in the skin called Meissner's corpuscles. They pick up discriminatory touch. It sends it toward the brain and spinal cord. If this is the spinal cord, the interneuron gets it, which is not only relaying up to the brain, but it's also connected to the motor neuron. And if that's, like, let's say that's a sharp pinprick, well, maybe that's going to fire that muscle to contract against it before the brain decides what to do about it. Called reflex, and we'll talk about that again. Look, same nerve. Here's the sharp pin getting those nerve endings. It comes in so intense. See the bulge? That's called a dorsal root ganglion. Cell body, cell body, cell body, cell body, cell body. So it makes for a lump because of all the cell bodies and the bodies are wider. The information comes in, the inner neuron picks it up so intense that it fires back out through the motor neurons, and that muscle is going to contract to where that pin poked. There it is again, the reflex arc, the sensory comes in into the inner neuron, the motor goes out. All right, so going back to what I said earlier with the PowerPoints. For whatever reason, your PowerPoints that didn't start with the neuron, they started with the nerve glue, the neuroglia or the glial cells. Now, I'm going to highly encourage that when we're done with our lecture, that you go onto YouTube and watch Ali Astrocyte. Okay, she was the grad student that I showed you before. Before we go, I'll show you the thumbnail of her videos. She does a real good job of explaining the neuroglia. She doesn't have a video that I'm aware of for the ependymocytes, but which are these right here. Some people say ependymal cells or ependyma, but I say ependyma. Some people say ependyma. Who cares? It's the same thing. I'll tell you what they are. Not a big deal. The glial cells are the support cells. It turns out that in the brain and spinal cord, the glia are different than what's in the periphery. And this is why the optic nerve is part of the central nervous system, because there are 4 helper cells or glue cells in the central nervous system, and there are 2 in the peripheral nervous system. So earlier when I said, hey, if we see Schwann cells, we know we're in the peripheral nerves. But if we see oligodendrocytes, it's in the brain and spinal cord. If we see astrocytes, brain and spinal cord, microglial cells, brain and spinal cord, ependymocytes, also known as ependyma cells, ependymal, some people say. Those are only in the brain and spinal cord. But the satellite cells and the Schwann cells, those are only in the periphery. If none of that makes sense, let me try it this way. The neuron, again, is royalty. The king, the queen, the prince, the princess, whatever royal figure you want to make the neuron. That neuron does not feed itself. It is fed by other cells. That neuron doesn't dress itself. It is dressed by other nerve cells. It doesn't protect itself. It is protected by other nerve cells. It doesn't quaff. It's quaffed by other nerve cells. What is quaffing? You know, clipping its nails and its hair and looking all good and great. No, it's made to look good because of other nerve cells called neuroglia, the nerve glue. Can you imagine if you were so VIP that every time you left your house, you had an entourage, you had a whole posse of people, and you turn and they feed you with a spoon, and you hold out your arms and they put your jacket on over you, and then a fan comes up and your bodyguards beat them up. You know, this is the neuron surrounded by its neuroglia. And once upon a time, scientists thought that these cells were just there to hold the neurons together. So they called them nerve glue. But they have discovered a lot, especially since the 1970s, of so much more of the importance of the neuroglial cells. And there are universities that are kind of in the forefront, and Stanford is a really big university on neurological advancement. So there's a lot coming out of Stanford University in California, and a lot of people that are really getting pretty well known in the world for their studies with neuroglia. They're Stanford people. And Ali Astrocyte's one of them, who studies under Dr. Mercola, not Mercola. She says in one of her videos what her research doctor's name is, who studied directly under some of these others. Let's see, this is YouTube. Is that one of Ali Astrocyte's videos? Yeah, maybe. Unless that's one of, okay, yeah. And I'll show you. The one that she does on the neuron itself is kind of annoying to me, because it was almost like her first attempt to do a video, and they wanted to do not only animation effects, but they tried to add sound effects, and the sound effects were almost overbearing as far as the music and the sound effects, which she kind of got away from with her other neuroglia videos. which makes the other neuroglial videos much more enjoyable to watch. But her neuron video, I don't know, you'll see if you watch them, you'll see what I mean. So, all right, so let me show you neuroglia. Let's see, do I have a picture of neuroglia on my stuff? I don't. So what I'm going to do is I'm going to go to the internet where I have saved some things. All right, this is an example of, on the left, neurons and neuroglia of the brain and spinal cord, and on the right, neuron and neuroglia of the periphery. This is a sensory neuron. The cell body is completely surrounded by satellite cells, and the axon surrounded by Schwann cells. But in the brain, this neuron is myelinated by oligodendrocytes. It's fed by astrocytes and ependymocytes, and it's protected by microglial cells. So notice that the neuron's got all the stuff plugged into it. This astrocyte right here, star cell is what it means. It's not only plugged into the ependyma, which is the lining of the chambers. The brain and spinal cord, as I said earlier, are bathed in cerebrospinal fluid. There are reservoirs of this fluid, and the membrane that surrounds the reservoir are made up by these ependymal cells. And so there's cilia on this side, which is kind of sweeping and moving the cerebrospinal fluid around the chambers of the brain and around the surface of the brain and around the spinal cord. But it's also able to permeate some of that into the cell membranes, and then the cell uses these processes to get cerebrospinal fluid and its nutrients either directly attached to neurons or attached to astrocytes, which take it and filter it and then feed the neuron. The astrocytes also attach to blood cells, and so it will take from the capillaries what it needs from the blood and then feed it over to the neuron. The neuron does not tap into the bloodstream. The astrocyte does, almost like mama bird feeding baby bird. Mama bird takes the food out of the blood vessel and then feeds baby bird. The astrocyte, the star cell, takes the material out of the blood and only gives the neuron what is effective for the neuron. It creates what's known as the blood-brain barrier. So certain things in the blood will never affect a neuron because it can't get through the astrocyte to do that. Another thing that the neuron is doing is forming a synapse with another neuron in the brain or spinal cord. And there's an astrocyte right there at that synaptic junction, making sure that the synaptic information flow is clean and moderated. And that's a very impressive thing. So it's not just neuron to neuron, it's neuron to neuron with an astrocyte medium, or if you will, monitor, to make sure that it's all good. Oligodendrocyte is a very complex sounding name, but let me translate all this to English real quick. Oligo as a prefix means rare or few. Dendra, if you remember, are trees. So these are cells with very few trees. And if you look at the oligodendra, there's a round thing. It's got 1, 2, like 4 branches sticking off of it. And each branch goes and then wraps around part of the axon to make the myelin sheath. So when the first scientists saw these under the microscope, they saw all these neurons with these branches, all these astrocytes with branches, all these ependymocytes with branches, and microglia with branches, but yet these other cells had very few branches. So that's how they referred to it, as the cells with very few trees, very few branches. And even though it sounds scary to say oligodendrocyte, if you know that that means few or rare tree cell, or not very many branches on the cell, I mean, that's pretty basic as far as names go. That's the neuroglia cell with the very few branches. And you only find oligodendrocytes in the brain and spinal cord, and their job is to myelinate the axons of neurons. If myelin is the clothing that the neuron is going to wear, then the oligodendrocyte is a neuroglial cell that dresses the neuron. The neuron doesn't myelinate itself. It's myelinated by oligodendrocytes. And what's even more interesting is this one oligodendrocyte, it's not just wrapping one neuron. It may be wrapping up to 25 neurons. One oligodendrocyte might have branches that end up surrounding 25 different axons of neurons for the myelination, which means that whatever it doesn't fill in, a different oligodendrocyte has to fill in the gap. So you'll see this one neuron right here with that oligodendrocyte, you know, they're filling in the gaps so that there's no point at which an axon is missing myelin, because the oligodendrocytes will move into position and remyelinate. And it seems that perhaps the reason why injured nerve cells in the brain and spinal cord don't repair is because oligodendrocytes, once they're set in place, if that neuron dies, there's no new oligodendrocytes to come in and fill in the new neuron. They're already preoccupied by the other neurons they're a part of. So that's different than in the periphery, where if the neuron is injured, new Schwann cells can form and then regenerate slowly but surely the axon. And it just takes some time. And it could be months to years before nerves in the periphery start to really repair well. And it's painful, but it can happen because Schwann cells work differently than oligodendrocytes. They're insulating that axon, myelinating it so that it has the fastest rate of speed of impulse conduction. Ependyma describes the membrane of the ventricles of the brain and spinal cord. All right, so it's the let's see if there's an actual etymology on it. Ependyma, EPENDYMA etymology. Integumentum ventriculum. So it translates to upper garment. Upper garment, or if you will, the outer garment that's worn. It's like an outer sheath, an outer layer of clothing. So when you look at the ventricles, it looks like it's got a little sheet of fabric surrounding the ventricle, keeping the cerebral spinal fluid in there. And so that's why it was called the ependyma, the outer garment. And again, it's Latin. So if you take yourself back to the 1400s, when Roman Catholic priests were describing things and they saw a thin lining of membrane, a thin cloth, if you will, along the inner part of the brain, that's what they would have called it. So these are the cells that link together and form this garment, this cloth. All they are a set of cells linked together to create a chamber for cerebral spinal fluid, which can actually permeate into the cells. And then their processes, these little branches, well, those would either connect to astrocytes, which then again take in the fluid material and then feed the neuron, or even go directly to the neuron. Now, microglial cells, these are bushy, full of little branches. They'll actually move around, keeping the area clean. If there's an infection, they will repopulate and they will branch out, and they'll actually fight the bacteria. So you shouldn't expect to see white blood cells from the blood in this area, because the microglia will do all that. But very important for the developing child, these microglial cells, their job is not only to protect the neurons, but to essentially break up connections between neurons that are not being used. When the brain is initially developing, neurons are forming, and they are so eager to connect to other neurons that they overly connect. And so one of the things that happens is when there are neurons that aren't really transmitting electrical impulses regularly because they're not part of the stimuli of the brain being, if you will, stimulated by the surroundings of the environment, these microglial cells will actually go around and they'll essentially prune or clean up unused synapses. And that makes room for future synapses. So it's a very good thing. You would think, oh, we don't want that to happen. You actually do want it to happen. You want any unused connections to be pruned and cleaned up so that in the future, as the brain develops more, as those stimuli of growth and development and new things, then there's more available connections to be made as the brain learns and learns and learns. So microglia just means small glue cells. Astrocyte means star cell. Ependyma cells are the lining cells of the upper garment surrounding cerebral spinal fluid. And oligodendrocyte means very few branch or very few treed cells. In the periphery, satellite cells are exactly what they sound like. They appear to orbit the body of the neuron cell. It's believed that they are essentially feeding the neuron, you know, oxygen, carbon, you know, some gaseous exchange and some electrolyte exchange, because the cell body is somewhat sequestered from the rest of the rest of its axon, the way the nerve develops. And so we believe that the satellite cells they're responsible for ion exchange. All right. But the Schwann cells, absolutely like the oligodendrocytes, myelinate the axon. Schwann was I guess a researcher or a doctor or somebody that journaled or initially described the nature of these cells wrapping around the axon in an effort to create a sheath that increases the speed of conduction. So these are what neuroglial cells look like. So again, here's the chart. These 4 to the left, they're only in the brain and spinal cord. These 2 to the right, they're only in the peripheral nervous system. So again, the astrocyte, it says maintain the blood-brain barrier, and that means it keeps things out of the blood that shouldn't affect neurons. But notice that it controls neurotransmitters around the synapses. Like I said, it moderates the synapse between neurons, and it regulates the ions, and it provides the metabolic support. It feeds the neurons. The ependyma, they line those ventricles. They are the shroud, the upper garment. And then, of course, they are also part of feeding the nutrients from cerebral spinal fluid, which is basically a clear, cerebral means brain, by the way, so brain and spine fluid. It's a clear fluid. It acts for protection, and it's got a little bit of sugar and oxygen in it, and it's good for feeding neurons. But myelinating the neurons, the very rare tree oligodendrocytes, and they are very good at myelination. But again, these microglial cells, they remove dead cells, they clean up injured areas. In fact, they can be overly aggressive in the inflammatory response, and it's you know, sometimes a bad thing, because it can cause, like, for example, with Alzheimer's and other, sometimes the microglia are over aggressive and they end up killing more neurons than they are protecting them. But their main job is to keep the area clean. They are the protectors. They are essentially the immune system of the central nervous system. You don't have to worry about the peripheral nervous system, because you've got a lot of blood with a lot of white blood cells in the periphery. But the blood is separated from the neurons in the brain so that it doesn't inundate the neuron with too much that would affect neuron function. But the satellite cells, again, we do believe that it's regulating, again, neurotransmitter release, the ion control, but they're probably the least understood as far as any of these cell types. And then Schwann cells, again, they are doing to the peripheral neurons what oligodendrocytes are doing, and that's making the myelin sheath, but much, much better at injury repair. I woke up from a dream once thinking that I was transplanting Schwann cells into the brain, and that was allowing the brain to heal. I don't know if it's doable, not yet anyway, maybe someday, one day. I've talked to a few people about it. I'm sure somebody's going to get the Nobel Prize because of my suggestion. It won't be me. All right, this is a real microscope picture of a neuron. That's the cell body. There's the nucleus. There are the dendrites. There's the long axon. Look at all these neuroglial cells all around it. Okay. That's why they thought it was holding the neurons together as glue. All right. So let's take a look at a more elaborate picture of what it looks like. Here is a blood vessel. The astrocyte in purple is all around the blood vessel, taking things out of the blood vessel and then directly feeding that over to the neuron. Here's the cell body, tree. Look at all these trees with their branches. Those are the dendrites. Look at the way the astrocyte just hooks right up into the neuron, not just the dendrites, but on the cell body. It's feeding material from the blood directly to the neuron. But hey, if it's something that could harm the neuron, this isn't going to feed it to it. So this is a complete barrier between the blood and the neurons, what's known as the blood-brain barrier. If you look at the ependyma, you see the cilia on one side moving the cerebral spinal fluid, and then you see the processes of the ependyma going over. And again, some of that would connect to neurons, some of that would connect to astrocytes, but it allows cerebral spinal fluid to be utilized by neurons. Here's an oligodendrocyte myelinating an axon. Here's an oligodendrocyte myelinating axons. And if you look in the background, we see the microglial cells, and they do move around. Again, they get really, really bushy. And then if there's a reason to fight a fight, they'll just mass produce more and more and more. They'll spread out and they will go on a feeding fest, protecting the brain from the pathogens of the world. All right, I'll put that down here. Let's get rid of that picture. All right, so here is a nice little set of slides that I found with the different neuroglia, just so that you can get a feel for what they are. It is not the major emphasis of this test by any means, and I highly doubt that any day you'll be doing massage going, I wonder how I'm affecting the neuroglia. But I do want to say that. One other thing with this picture and Einstein, and probably the reason why Ali Astrocyte likes astrocytes so much, is originally people thought, well, Albert Einstein must have a lot more neurons in his brain because he's so smart, right? Albert Einstein's brain didn't have any more neurons than yours or mine. But what they found was he had an absolute extraordinary amount of extra astrocytes. Not 10-to-1 neurons, astrocytes. The astrocytes in his brain were way, way more than expected. All right. There's 10 times as many neuroglial cells as neurons. All right. So not, I said about the same before. No, 10 times as many. That I've seen that before. I think that's right. So if you have 100 billion neurons, you have 1 trillion. Isn't that 10 times more than 100 billion? So then you have 1 trillion neuroglial cells in your brain. And there are 4 types. The astrocytes, which are the star cells, shaped like stars, that's what astrocyte means. They are great at feeding the neurons. So please, when you get a chance, take a look at this slide. Look at the way they're abundant, they're versatile. They do buffer or moderate the synaptic transfer. They're very important for feeding the neurons the good stuff from the blood. That is astrocytes. And again, Einstein, for whatever reason, had a lot more astrocytes per expected compared to neurons. So it seems to me that those astrocytes made his neurons work very effectively. Those are astrocytes. The microglia, again, they're smaller in size, but they are the protectors. They are the bodyguards. They protect the neurons from pathogens, from debris, harmful substances. But early on in life, they are important for making sure that synapses are functional and useful by pruning unused synapses to make room for future synapses. As far as ependyma, again, if this is a canal and there's cerebrospinal fluid in here, the ependyma are the cells that line the canal, and then they will feed the neurons on the other side of the canal the benefits of cerebrospinal fluid. The cilia moves the fluid so that it's constantly in circulation, so that we can keep the cerebrospinal fluid free and, if you will, refreshed. It's not old cerebrospinal fluid. It'll circulate around the brain and spinal cord and then be sifted off so that all new cerebrospinal fluid keeps on bathing the brain and spinal cord. The ependyma is a way to get the benefits of cerebrospinal fluid into the neurons. As far as the neuroglia themselves that are oligodendrocytes, here we see an oligodendrocyte myelinating that axon, that part of the axon, that part of the axon. But look, even this neuron's axon and that neuron's axon. So it may not have nearly as many branches, or at least the scientists that first saw them didn't think it had nearly as many branches as some of the other glial cells. And so it may be probably improperly named few or rare branched or treed cells. But we definitely know they are what make the myelin sheath of the central nervous system neurons, including in the optic nerve. As far as the periphery, the satellite cells that surround the cell bodies versus the Schwann cells, which form the myelin sheath on the axons of the peripheral nervous system. All right. So I do believe that is our stop point for today. Let me just check and see if I'm right. All right, parts of the neuron. And there's that afferent efferent thing again, and the reflux arc. So yeah, so that's where I'll pick up on tomorrow at 3. Let me go ahead and stop the share here. Let me get out of these PowerPoints first, and I will stop the share so I can see everybody. All right, so hopefully today's discussion of the nervous system was a good introduction as far as central nervous system versus peripheral nervous system, and a good discussion of the cells, the neurons, and the neuroglia that make up the greater portion of our brain, spinal cord, and nerves. So we also want to include blood vessels in that, and that's actually part of the cardiovascular system. But again, it's very important for all of the nervous system to be supplied with blood. When I see you tomorrow at 3 p.m., we'll continue where we left off, looking at those neurons and how they form the reflex arc. If there are any questions, please let me know. In about a half an hour or so, I'll send you an email with today's recording and link. And Ina was nice enough to include her notes in an email to me, which I will pass on, which has Thursday's information for those that might have missed it. All right. So have a great rest of your evening. Enjoy the day. It's a really nice one out there. And I'll see you tomorrow at 3. Bye. I'll reply to Dr. Annie's email tonight with today's notes. Oh, fantastic. Love it. Thank you.